Okay. So now we're here. So we're starting with chapter 2. Chapter 1 should be basics. Chapter 2, chemistry. We had the different atoms and the different bonds. We just started talking about them. Covalent bonds, you are sharing your electrons between two atoms. Each of those atoms is getting at some point to see those electrons. With ionic bonds, you are actually donating or transferring that electron permanently. In these, if you are having donation or loss or gain or whatever you want to call it between electrons, then you have to have redox reactions as well. So anytime you have an ionic bond, you also have reduction or oxidation as well. Hydrogen bonds, what happens here, these are of course more complicated. You have the electronegative thing, which often is oxygen, right? And it hogs electrons. So it's going to tend to have a negative charge. And then it is covalently bonded to that hydrogen. But because that hydrogen isn't seeing that electron as much, it has a positive charge. I'm just going to write this as positive charge because I don't want you to think that this is the same thing as this, because this is not the same thing as that. This guy has a positive charge, but still gets its electron. This guy completely lacks any electron. Okay, so those are not the same thing. I want to be very clear about that. I want to erase for just for a second, so I can get rid of this. So I can keep talking about my hydrogen bonds. All right, so back to those guys. So then when we have this positively charged hydrogen, this guy is going to be more attracted to basically what is that electronegative on something else. So electronegative on attached to some other thing, molecule X, it doesn't matter. But this guy has that slight negative charge. And then that negative charge with that positive charge. This is the H bond. That attraction is. So with hydrogen bonds, the hydrogen bonds themselves, that H bond area, there is no electron exchange or anything going on in that bond. There's no even hogging or anything in that bond itself. That's just an attraction that occurs because of other atoms hogging in their own molecules. And so here the hydrogen bonds are created from the polarities that result from that. So for it to be a hydrogen bond, it has to have a hydrogen that is positively charged involved in that attraction. So those three, the covalent, the ionic, and the hydrogen bonds are our biggies for our chemistry bonds. Okay, so how do molecules interact with water and explanations that we discussed? This is all those terms dealing with hydro. You can be phobic or philic. If you're phobic, you're water fearing. And that's because you are nonpolar. Philic, water loving. And that is because you are polar. And where do we get polar and nonpolar from? That crap. From our hogging situations, right? And so we know that oxygen and hydrogens, these guys have their positive charge, and this guy has its negative charge, and then this guy is going to be attracted to the positive charge of this, neg, and whatever, you get that. And so what would that make this right here? A hydrogen bond, right? So anyways, this polarity aspect of water makes other polar molecules interact with it very well. If you're a non-polar, you don't get along with these positives and negative polarity guys, and so you don't mix well. So non-polar, hydrophobic, these are like our oils and our fats. they have this long hydrocarbon chains. And the hydrocarbons are important because the carbons are very, very stable when they interact with the hydrogens. They share very fairly with the hydrogen. So nobody has a charge, nobody's hogging. It's not like with oxygen. So those long hydrocarbon, there's the word, chains, those guys don't have any charge. They're going to be nonpolar. They're going to be hydrophobic as a result. We also have these molecules that are called amphipathic. And this means that they have both of those things going on, sorry. So this would be things like phospholipids, where you have that phosphate group that is very negatively charged. And then you have that hydrocarbon tail that is the hydrophobic part. So that's amphipathic. Phospholipids are incredibly important for the structure of our membranes, and that's what gives us our selective permeability and keeps all of our compartments compartmentalized and gives the cell basically its ability to function as a unit. So don't underestimate the power of those amphipathic molecules. All right, so those are our little explanations for that. For our pH and what's being measured there, remember with pH, you are measuring the negative log of a concentration of hydrogen ions. So these are proper hydrogen ions. And acids mean more H plus, base less H plus. And because this is a negative, this is what creates that inverse of the number. So more H plus equals a lower pH number, so towards 0. And then less H plus equals an increased pH number towards 14. And then neutral, we have a pH of 7. So on that 0 to 14 scale, you could see it as being, and as you go towards 0, you have excess H plus. As you go towards 14, you have a lack of H plus. So whenever you have equal H plus, you don't have too much and you don't have too little, it would be right smack in the middle at 7. Okay. You just need to remember that it's an inverse ratios that we're talking about here with that negative logarithmic scale. So remember that your acids are 0. Remember that your bases are towards 14. Remember your neutral is 7. And then remind yourself that because this is an inverse, the 0 actually means more H pluses. And the H of the pH is literally referring to the H pluses. All right. What makes a molecule organic? You have to have carbons and hydrogens in the backbone. You need both of them. Large molecules and their parts. For these guys, we're talking about our macromolecules. So this would be things like proteins. These are made up of amino acids. We would have nucleic acids like DNA and RNA, and these are made up of nucleotides. And a nucleotide consists of a 5-carbon sugar, ribose or deoxyribose. It consists of a nitrogenous base, ATCG in the case of DNA, or AUCG in the case of RNA. And then we have our phosphate group. So that is a nucleotide. With our carbohydrates, we have monosaccharides, which are just single sugar molecules that come together to make up a carbohydrate. And with our lipids, we have various forms of lipids, like we have fatty acids, where we'll have like a charged head, like a glycerol group with those long carbon chains. We have technically our phospholipids will fall in that group. We'll have our sterols, like cholesterol, that help give support to the membranes and give structure to those guys. For the protein structures themselves, remember that we have 4 different levels of structure for our proteins. We have the primary structure that is just the amino acid sequence. So that could be alanine, 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 glutamate, alanine, phenylalanine, phenylalanine, whatever the sequence is of the 20 possible amino acids. Just the literal sequence of it is your primary. Your secondary structure is once those, alanine, alanine, alanine next to each other start to interact with whatever their polarity is, whether they can make hydrogen bonds, whether they are nonpolar, or whether they have straight up ionization capabilities or can make double bonds, whatever the thing next to you can do. Now we start forming these, that was weird. We start forming these basic structures of alpha helices, which are just spiral structures, or beta pleated sheets, which are flat segments that go back and forth, like a pleated dress or something. So those will happen from the amino acids immediately next to you, starting to create those structures. That's the secondary. The tertiary is once we have those structures formed from those direct amino acid to amino acid relations. Now we can start folding up the whole structure, tying your bow, if you will, right? Creating that whole structure from that whole ribbon sequence, and having things interact across the way from one another, creating disulfide bonds from amino acids that aren't anywhere next to you at all. They're across the whole sequence. And these things start to form up to make larger folded structures. That's your tertiary structure. Then when you can take your multiple bows that you've now folded and put them together to make a multi-bow functional protein, that's your quaternary structure. Okay, so we're going to move on to chapter 3. In chapter 3, we have our 5 I's: your inoculate, where you're introducing your microbes; your incubation, where you're growing; your isolation, where you have your pure cultures of your microbes. You have inspection of your growth, looking at different color, can you grow on this media? Can you not grow on that media? Did you make when you make ran into the wall? Sorry, I'm very clumsy. When you make inspecting color changes, that's what I'm trying to get at. And then you have your color changing media, things like that's your inspection. And then using all that information, whether it grew or not, color changes, what it looked like in microscopes and whatever, to identify. Those are your 5 I's. Then we have the different types of media and how we used those. Here we're talking about selective media that has something in it to select for certain bacteria. Usually it helps some things to grow and other things will be prevented from growing. That's our selective. Differential, we usually see color changes that are going to be resulting from metabolic byproducts interacting with chemicals in the media, cause color change. Then we have media called enriched media, where we have added growth factors that the bacteria can't make for itself and has to have provided in its diet. That's like our vitamins, for example. So we can't make those and need to be provided in our diet, just like bacteria have their own growth factors. Bacteria that need growth factors are called fastidious, and the media that they grow on it's called enriched media. As far as isolation goes, we have, everybody at this point probably knows what a quad streak method is, and that we use it for isolation. A pure culture is when you take one colony grows up from one single cell. So a single colony, if you can get one isolated, you can create a pure culture from that. So it's one single species from one colony. That's what a pure culture is. For microscopy, we need to be able to at least have a general understanding of how microscopes work and their parts. Be aware that we create our real image from so describing the microscope and how the light flows, I feel like helps understand what each of these images are created as. So the light comes through the light source through the condenser and shines it onto the actual specimen, and then hits the first lens. That's your objective lens. That creates the real image. That's the very, very first image that gets created. So that's the real one. It shoots through some mirrors and another lens, which is the ocular lens. So we've now been manipulating that image again. So now that's the virtual image that hits the eye. Okay. So that's the images that are created there. Oil, we will use oil that is to reduce light scatter. and be able to increase the clarity of the image and be able to see more detail there. It doesn't do anything else. It doesn't lubricate it. It doesn't keep the species alive or kill it or anything like that. It's just reducing light scatter. Stains, we use those for differentiation as well as to improve contrast in our cells so we can see the structures of the cells more clearly and identify certain structures using our differential stains, like the acid fast stain that we actually will be using to determine if your organism has mycolic acid. If your organism has mycolic acid, it is positive for the acid fast stain, and it must be a mycobacterium species. The endospore stain that determines if you have endospores or not. Only gram-positive bacteria can have endospores. We learned that in the lab. And only Bacillus and Clostridium species make endospores. So those guys are gram-positive. And so we can use that stain to differentiate for that. We can also look for presence of things like capsules and that sort of stuff. So that gives us a little bit of leeway into chapter 4, our bacteria and our archaea. Bacteria and archaea, these guys are eukaryotes. They do not have a nucleus and they do not have organelles. A nucleus is an organelle, to be clear. It's the organelle that houses your genetic information, and that's what it does. It's essentially like the information storing, the CPU, if you will, of all of the cell. But the DNA never leaves the nucleus. It always stays there. All of your genetic information is housed in your nucleus. And so your DNA is kept there. And if you want to make things from that DNA blueprint, you want to make a copy of that one gene, then you will make your ribosome, or you will make your RNA. Oh, geez. You'll make your RNA copy, your messenger RNA, which will be read by the ribosome and translated into protein later on. But all that DNA is kept in the nucleus. Your organelles are structures in the cell that have specific functions in the cell. And these guys are, let's go over them. Let's do it, right? So we'll get back to, I said about the organelles. Let's talk about that in chapter 5. Let's just stay on chapter 4 right now. I don't want to get confused. Chapter 4, these guys don't have organelles, which are structural, like structures that have specific functions in eukaryotic cells. These guys still have those functions, though. I think people get lost on that. They still make energy. They still make ATP. They still make proteins. They still do all of these things that we as eukaryotes do in our organelles, but they just don't do them in organelles. They just do them either in the cytoplasm or along their cytoplasmic or internal inside membrane, if they have two membranes, if they're gram-negative. So that's important to remember. They're still doing those processes. They're just not compartmentalizing them. All right, so gram-positive, these guys have that thick peptidoglycan cell wall that gives them that extra structure and support, especially in high osmolytic pressure environments. So like high salt concentrations. Gram-negatives have a thin peptidoglycan layer, but they have an outer membrane that helps support their structure. We also have others, right? And that kind of brings us down to that bottom point, the groupings based on the acutes. Firmacutes have that firm, thick cell wall. Those are our gram-positives. That means gracilacutes, our next G, will be our gram-negatives. Teneracutes, also think of tender. So like if they're tender and they're soft, they don't have a cell wall. That's the teneracutes. And then Mendozacutes, like my old boyfriend, Joey Mendoza, from a long time ago, it's archaic. Mendozacutes are archaea. They have cell walls and cell membranes, but they're not made up of peptidoglycan or anything that we would recognize as normal. They have some weird organic hydrocarbon stuff going on. All right, so our cell shapes and our arrangements. We have cocci that have the round shapes, the bacilli that have the rod shapes. Our arrangements, staphylo is going to be our grape-like clusters. Strepto are our chains or strips, right? And you can have palisades where you have your fence post arrangement of rods kind of next to one another. So that's our cells and arrangements, and I will ask you about those on this test. Yes, it wasn't just for the lab stuff. Flagella and movement. So bacteria can have flagella, not all bacteria have flagella. And flagella are the only ways by which bacteria can move. And they move in a rotating like motion. So the flagella actually rotates. It doesn't move in a whip-like fashion. And so they can either rotate it clockwise or counterclockwise. If it's running, then it will be rotating in one direction. So it's a run, right? And it'll go straight. When it switches direction from like clockwise to counterclockwise or vice versa, then that causes a tumble. And they will use a series of these runs and tumbles in order to direct their movement either away from or towards a chemical or a light source. That's chemotaxis or phototaxis, respectively. They have different flagellar arrangements. We have monotrichus, where you have one flagella only. You have peritrichus, where you're covered all over with flagella. You have lophotrichus, where you have a tuft or tufts of multiple flagella, but they're in little tufts or groupings. And then you have amphitrichus, where you have flagella at each end of the bacterial cell. So those are our flagellar arrangements. Capsules are primarily going to be saccharide based, so sugar based. Maybe even proteins can be involved in it as well. But either way, it's like a sticky coating that they have that allows them to attach to surfaces. Now, capsules are pretty densely associated with and closely associated to the cell, whereas slime layers are similar molecules, but they are loosely associated. Both of these guys can allow for formation of biofilms, where we'll have several different species of bacteria kind of stuck up on one another on a surface, creating a literal film. And their products from just living send signals to the cells nearby, letting them know about their environment. And that communication method is called quorum sensing. Then we have the ability to evade phagocytosis as part of having a capsule as well. It's hard for our white blood cells to eat up or phagocytose our bad guys when they have a sticky coating on them, though our immune system has found ways to work around that as well. All right, next is endotoxin. Endotoxin is also known as LPS or lipopolysaccharide. This is a molecule on the surface of gram-negative outer membranes. Only gram-negatives have this, and all gram-negatives have this. It is a PAMP, right? And we'll talk about that a little bit later in our review. But a PAMP is a pathogen-associated molecular pattern. It is something that our body immediately recognizes as bad guy and initiates our inflammatory response. So that's kind of the importance of endotoxin for our purposes. Endotoxin, because endo being inside is part of the actual bacterial cell. Other toxins that bacteria make tend to be exotoxins. So they'll actually secrete those toxins, things like cholera toxin or tetanus toxoid. Those things they actually secrete. So those are exotoxins. Plasmids are little extra bits of circular DNA that have extra genes on them that can help with survival, but the cell could technically still survive without it. So they're not required for survival. They just can make it so that the bacteria is either more pathogenic or is more likely to survive in given conditions, like it can give antibiotic resistance or production of toxins. Endospores, again, these are hardy, dormant structures. They're always dormant. They do not multiply. They don't do anything at all except for wait for conditions to improve. When conditions improve, those hardy structures will go through something called germination to create the vegetative, metabolically active, reproducing versions that we usually think of when we think of cells. So that's the vegetative cells that are actually multiplying, causing the infection, producing toxins and things like that. So vegetative cells do that. When the vegetative cells go through hard conditions, then they will go through sporulation and become sporangia, creating their own endospores. Each cell can only make one endospore. So it's not a reproductive method. Groupings based on, we already talked about the cutes. So moving on to chapter 5. Again, stop me if there's something you guys want me to cover more, okay? Otherwise, I'm just cranking through. Chapter 5 was our eukaryotes. Eukaryote means true nucleus. So these are cells that have true nuclei and therefore have true organelles. At this point, I would hope that you guys would know the difference between the eukaryotes and the akaryotes. We think the eukaryotes came around from some basic akaryotes that like one ate another one and they got benefits from one another. And that symbiotic relationship ended up being a permanent reliance, and that those little eaten up guys that helped us out whenever we ate them, became our organelles. And there's evidence to support that the mitochondria and the chloroplasts have their own circular DNA, just like bacteria have. They have double membranes like gram-negative bacteria have. They divide separately from the cell. They have their own types of ribosomes that are bacterial ribosomes and not our own ribosomes. They basically, like I said, they divide on their own, and they have all the genetic information to exist separately from our cells. And our genes do not code for them. They have to code for their own existence. So it's pretty interesting, pretty strong support for that concept. So our organelles and their functions. Organelles, again, are basic structures in the cell that have essential functions within the cell. These processes still occur in our eukaryotic cells. They're just not compartmentalized in eukaryotes. So we just compartmentalize them in the eukaryotes because we have a lot more going on as multicellular organisms. So our organelles will include things like the nucleus that houses your genetic information, like the endoplasmic reticulum. You have the rough ER that's studded with ribosomes that's important for protein production. And you have the smooth ER where we have production and adding on of non-protein molecules to other molecules that we're making. Then we have the Golgi apparatus. The Golgi apparatus is for packaging and transport of vesicles. This includes making things like our lysosomes that contain our lysozyme and toxic oxygen products and things like that we will degrade our bad guys with. Then they will also help export anything that needs to be exported out of the cell with those vesicles. The mitochondria create our energy powerhouse where our aerobic respiration occurs. Then the chloroplasts are where we have photosynthesis occurring. And those are like major organelles and their functions. Our flagella for the eukaryotes do move in a whip-like fashion, but they're also membrane bound. So our membrane is continuous over those, and they're made of microtubules and all that. So eukaryote cells can move via flagella, but they can also use cilia for movement. And they can also use pseudopodia, which we learn about those guys from our protozoa. Mitosis and peeing on a mat. So PMAT. So in mitosis, which is where we actually duplicate our cells, is how we make copies of our cells, especially as multicellular organisms. Bacteria go through something called binary fission, but we go through something called mitosis. And this occurs in phases, PMAT phases. P is prophase. Here we're just consolidating everything, basically. Then M is metaphase. We're going to line up everything that we consolidate. A is the anaphase. We're going to pull it apart into the separate areas. Well, yeah, pull apart or separate. And then T is telophase. Or we're going to be totally done. We're going to separate these guys completely into different cells. So that's really the gist of what happens in mitosis and the major steps that are involved there. I'm not going to show you pictures and have you identify the different stages of mitosis. That's not what microbiology is for. That's genetics. So where is energy made in eukaryotes? Like I said, eukaryotes make their energy in the mitochondria. So where would we make energy in prokaryotes, though? They don't have mitochondria. So they have their whole aerobic respiration, glycolysis, and that stuff happens in the cytoplasm. But once you get into the electron transport chain where we're shuttling those electrons through those membrane-bound cytochromes, those proteins, that electron exchange and then pumping the hydrogens across that membrane all happens on that inner or cytoplasmic membrane of bacteria. So they still do it, they just do it on their inner membrane. Chloroplasts, again, these are structures that are important for photosynthesis, which we'll review in a little later chapter. The organisms that are eukaryotes that have these are going to be our plants and our algae. Fungi do not have chloroplasts. All right, fungi, again, these guys can be opportunistic pathogens. We don't have a whole lot of them that are true pathogens. Most of them are just opportunistic, so they can be part of like your normal flora, or they don't normally cause illness except in extreme, like you have lowered immune system or you're already exposed to some other illness or something like that. They do form spores, but their spores are for reproduction. They're nothing at all like an endospore. So endospores are dealing with bacteria. I will always call them endospores. Spores for the fungi, I'm usually going to refer to them in the context of them being for fungi reproduction, or I'll call them like parts of like conidiophores or something like that. Those structures that we learned about being part of the fungal structures for reproduction. So whether they are covered or uncovered, the uncovered ones are the conidiophores. So sporangiophores are the covered ones. And we'll remember that because the lab had like penicillin and aspergillus. Both of those guys were uncovered conidiophores, right? So those are spore forming asexual reproduction. Oh my gosh, for the fungi. If you're algae, those are our primary oxygen producers, the algae and plankton that are photosynthetic. They are primary oxygen producers on the planet by far, not the plants, even though we see the plants because they're macro. I can assure you that most of our oxygen in our environment was made by our microbes. Other than that, we don't care much about the algae because they don't cause disease, and we're mostly talking about disease. Protozoa, however, we have the different kinds of protozoa that can move by flagella, by cilia, by pseudopod, or by not moving at all. They're amodal. And that's how we characterize our protozoa. They're all single cell, and they're all of the non-modal ones are going to be the parasitic ones. You can have the other ones that can be parasitic too, but literally all of the non-modal protozoa are parasites. This will include things like malaria, which is non-modal, or we could have giardia, which is has flagella, right? And that's still going to be a pathogen, but not all flagellated protozoa are pathogens. That's what I'm saying. So there's the protozoa. They're all single cell though. Then we have the helminths. These are our multicellular parasites. So these are our parasitic worms. This includes all of the helminths. We have the roundworms, which are nematodes, our flatworms, which are platyhelminthes, and then the platyhelminthes are subdivided into the cestodes, which are cestodes. Our cestodes, those guys are tapeworms, and then our trematodes are our flukes. All right, moving on to viruses. We should know about the basic structure of our viruses, that they have genetic material of some kind. What kind of genetic material can they have? Well, they can have DNA, they can have RNA. That DNA or that RNA can be single-stranded or double-stranded. And if it's single-stranded, it can be positive sense, meaning that it would be ready to read by ribosomes, or it can be negative sense, meaning that we need to make the copy, right? The A to T or C to G copy of it in order to read it. That'd be negative sense. So you can have 6 different kinds. DNA that is single-stranded positive, single-strand negative, or it could be double-stranded. Or you could have RNA that is single-strand positive, single-strand negative sense, or it could be double-stranded. So 6 different kinds of genomes there. They also have to have a capsid. Capsid is just a protective protein coating that covers that genetic material. All viruses have a genome and a capsid, all of them. That structure together is called a nucleocapsid. So you could have a nucleocapsid. So here's your DNA, and maybe it's single-stranded, I don't know. And then you have your protein that coats it all around it to protect it. This is the protein. So these guys, this whole protein circle structure is called the capsid. And then we have the genome here, whatever that is. Could be RNA or DNA. Could be double-stranded or single-stranded. And that could be plus or minus. Okay, so that's the gist of that part. And then this whole structure here that has the capsid structure that's wrapped all around it, right? And the genome, this is called the nucleocapsid. All viruses have this. These individual single circles is called a capsomere. These are the segments that make up the capsid that wraps around the genome. And around that, you could have a stolen membrane. And if you do have that stolen membrane, this equals the envelope, the viral envelope. Not all viruses have that. All viruses have a nucleocapsid, so the capsid coding the genome. All viruses have that. Not all viruses have a stolen membrane. If you do have that stolen membrane, then on the outside, you have to have your spikes. Now, if you don't have that membrane, you still have spikes, but it's going to be part of your capsid, right? It's not going to be studying anything outside of that capsid, just the capsid itself. But if you have that envelope on the outside, then your spikes to get into the next cell has to be on the outside of that. And so we'll stud that membrane with the spikes and then push through that studded membrane with our nucleocapsid and coat our nucleocapsid with that blanket of the stolen membrane studded with the spikes. So then you would have a structure that looked like this. So you have your thing, your nucleocapsid that's got its little protein coating, and then you will bud out and then pinch off, and this will have all your spikes on the outside of that membrane. So that will show your envelope with the spikes on it and your nucleocapsid that's on the inside, and that's the structure of the virus. With the life cycle of the virus, the 6 steps that are involved, you have adsorption. This is attachment. Then you will have entry, basically. So you will have, what do I want to call this? The terms. I want to be sure I'm using the right terms for you guys, not just explain. Penetration is the word that I use. Penetration. This is entries. This is what I was getting at. You'll have your uncoating, which usually happens at roughly the same time as the penetration. So this is just removing that envelope or the capsid and then allowing us to have access to the genome. So removing any coding on that genome so we can actually start using the genome. Then once we have access to the genome, we can go through synthesis. So this, we're just making our parts. So we're making the genome copies and the capsid parts. So the capsomeres. And then building up our virus, that's the assembly step. And then the last step is release, which we just talked about the budding if you do bud. If you don't bud, for example, the polio virus doesn't have an envelope. It doesn't bud. So what it does is it gets exocytosed. So just like endocytosis, where the cell's going to eat up something, exocytosis, sort of like it vomits it out. So it puts something in a vesicle, like it would through the Golgi apparatus, and then merges it with the membrane to sort of like push it out of the cell, like you would secrete anything else, like waste or something. All right, talking about cytopathic effects, these are just any negative effects of the cell as a result of being infected by the virus. You're not using your cellular machinery like it should be, and all we're doing is cranking out virus parts. This can also lead to like overproduction of the spikes on the outside surface of cells, causing cells to merge together and forming syncytia, or it could be enlargement of large inclusion bodies and damage of organelles and things like that you can see in very clearly in microscopes, as well as being expressed as effects of the cell causing pathogenicity in the host. All right, moving on to latency, lysogeny, and all of this BS. Latent viruses are viruses that are hiding out in some form or another in their host. So anytime that you have genetic information from a virus in your cells, but you're not actually making active viral pathogenesis occur, that is latency. When you have latency in human hosts, we just call it latency. When you have latency, when you're a bacterium that is infected with a bacteriophage, which is the virus that infects bacteria, then the latency is called lysogeny. And that's the term for whenever that prophage, which is the genome of the phage, gets incorporated in the genome of the bacterium. So the prophage and the bacterial genome become one. That is lysogeny. And sometimes genes from that bacteriophage can give new traits to the bacteria that's infected with it, and that is called lysogenic conversion. This can actually become semi-permanent in a sense that whenever we look at things like Streptococcus pyogenes, most of its pathogenic traits come from genes that are lysogenized genes. So it got those toxins and things and stuff that it makes from the genes that it got from having a bacteriophage. And it's true for a lot of our bacterial diseases are from lysogenic conversion. The lytic phase for bacteriophage, again, lytic and lysogeny, these guys have to do with phages. So this is bacterial viruses. When we talk about latency, we're mostly talking about latency and proviruses. These are eukaryotic viruses. Whereas prophage, lysogeny, lytic, these are temperate phage features. So a temperate phage is a phage that can switch between lysogenic and lytic phases. Lytic phase is whenever you're making viruses and they're busting out a bacterial cell, like what you typically do with a viral infection. Lyse the cells open your new bacteriophages. They don't do exocytosis or anything. They always bust out, always. All right. Provirus is the same thing as prophage, but with a provirus, we're talking about viruses incorporating their DNA in like a human host, for example, not in bacteria. Right. So we can have viruses that can hide out in our DNA as proviruses, like we have with HIV, for example. We have viruses that can just hang out inside of cells and become latent, like what we see with like tuberculosis, where it just hides out in your alveolar macrophages. And we can also have infection with bacteria that have been lysogenized by bacteriophages, temperate phages, and get extra genes that make them pathogenic, and those can affect us as well. All right, so moving on to prions. Prions are literally just misfolded proteins. That's all they are. It's a protein that has a normal function in the body, and it becomes misfolded, and then other healthy versions of that protein will come into contact with it and want to misfold also. They aggregate together, and those aggregations cause holes to form in the brain tissue, leading to spongiform encephalopathies. All right. Unit 2. Genetics. All right. So phenotype and genotype. Phenotype, let me make this big again. Not that big of this. Phenotype is expression. That's what you actually see. Genotype are the genes that you carry. So if you carry a gene for green eyes and a gene for blue eyes, but you yourself have blue eyes, your phenotype is blue eyes, but you have genes for green and blue, right? So that's your genotype would be explaining all of everything that's going on, even the stuff that isn't expressed. So if we're ever talking about what is actually being expressed, and I'm actually asking you about what's being expressed, even if I give you the genetic information, if I ask about what's being expressed, that's talking about the phenotype. All right, so DNA and RNA and the nucleotides, we already went through this in the chemistry one, so I'm not going to talk about that part of it. Do know that DNA is deoxyribose 5-carbon sugar. RNA has ribose 5-carbon sugar. DNA has A, T, C, G, adenine, thymine, guanine, and cytosine, whereas RNA has a U, C, G. The U is uracil. That's the nitrogenous bases for those guys. So those are the differences. We usually in eukaryotic cells, we don't see double-stranded RNA ever. So that can actually be a PAMP. So if we see double-stranded RNA in our body somewhere, we can see it as a PAMP. It's an automatic signal that there's a viral infection. When we're replicating our DNA, that's just making copies of DNA. DNA replication only refers to making copies of the DNA for like mitosis purposes, right? And so DNA is a double-stranded helix and it runs like a ladder where you would have A here and T here, and that's one part of it. And you have C and G, and that's another part. And then maybe G and C and whatever, and you get it. So this would guys would be like this, all the way across, all the way down. Now this backbone here, the backbone consists of sugar phosphate units. So it's a sugar bound to a phosphate, and then another sugar bound to a phosphate, and another sugar bound to a phosphate over and over and over again. So we could go down this whole row and look at it like that. I need to space this out a little bit better if I'm going to draw it. But this direction of this sugar phosphate, if this sugar phosphate, sugar phosphate, sugar phosphate, then this other side might be sugar phosphate, sugar phosphate, sugar phosphate, and whatever. So that the end down here, you would have sugar phosphate. What starts over here? Is it a sugar or phosphate? A sugar. What's down here? Is that a sugar or phosphate? A phosphate. And I feel like this is obvious, but this is how I'm going to dictate to you guys. This is what I'm talking about with the 5' to 3'. At this end, that 5' end is what's exposed because there's no phosphate at that end. So it's going to be a 5'. 3' is what you're seeing down here at this end with this phosphate group. So the pre-prime sugar is the one that's the carbon that's free on the sugar because the other one's got that phosphate on it. So this is 3'. Everything is read in the 5' to 3' direction. And you're just going to have to remember that it's always 5' to 3' direction. Everything operates in the 5' to 3' direction always. So if you're going to add something on, it has to be from the 5' to the 3' end. So when you add something on to something from the 5' to 3' end, you would be adding on a sugar phosphate. So now you have the 3' still at the end, right? That's what I'm saying when we talk about that, building on. Anyways, so that is the reason that we have these different directions facing on these backbones. And this is what we call anti-parallel. This top strand here is going in a different direction than this bottom strand here that is complementary to it. That's anti-parallel. Whenever we make copies of these guys, we will separate them. And make copies from the original. So this is the original from the top one up there. This is the original from the bottom one down there. And then so you would make your copy matching up your ATCGs. So now we've got one old and one new. So this aspect of DNA is called semi-conservative. Okay. So that's those terms. Now, whenever we are actually breaking apart our DNA, because it is so long and we can't just start at the end and work to the end because those ends are often tied up in things like telomeres and extra sequences that accommodate for loss that we will see every time we duplicate our DNA. We have to start at somewhere called origin of replication. And so we'll open up that DNA basically as a bubble. And that's our origin of replication. So we have our single nucleotides here. And we will, again, be moving where our new piece will have our 5' and we'll be going towards in to the 3'. And we can just push this fork open. This is our replication fork. And this will open up. But the other side, in order to make a copy that matches on with it, has to start here and go this way. Because remember how these strands are oriented? If that is going that way, then this would be 5, and this would be 3, and this would be 3, and this would be 5. And in order to make this be 5' going in the direction to 3', we have to start at that where we're opening from. And so this means as we open this more and more, and this becomes open and we can keep going on in and in and in, then we can make another segment that matches up in here. These are made in fragments, and these little fragments is the Okazaki fragment. So that's that guy there. And the Okazaki fragments have to be sealed together by DNA ligase to make them one long continuous strand. Whereas the leading strand, the one that can just push through 5' to 3', just straight on through, is the leading strand. And this is the lagging. Lagging strand is made up of Okazaki fragments. Those fragments have to be sealed together to be one continuous thing by the DNA ligase. Whereas the leading strand is just one continuous piece. It doesn't need any sealing together. And so that's how DNA replication will go on. Now, that's just to make copies of the DNA. That is just DNA replication. If you want to talk about transcription and translation, you're talking about looking at products of the genes. So replication is just making copies of the genes. We're not doing anything with the products. If you want to make products from your code, you are going through transcription and translation. Okay, so I'm going to clear this screen now so we can talk about these things. So transcription, we are taking our DNA gene and turning it into a messenger RNA copy. This is our working copy. All right, so we only need a copy of that one gene. We don't need a copy of the whole dang everything in your DNA. We just need that one gene copy to make that one product. So what we'll do is go through transcription, which involves the 3 main steps of initiation, elongation, and termination that a lot of these things have. We will initiate at the promoter sequence, and that is where your RNA polymerase will bind on and then do the whole normal 5' to 3' thing. You make a single strand RNA copy of the messenger RNA that can be read by a ribosome. So that's transcription, is making that copy. Translation we're going to take that messenger RNA copy and turn it into a protein, which is just an amino acid sequence using ribosomes and tRNAs or transfer RNAs. All right. And basically in translation, what's going to happen is the ribosome binds onto that messenger RNA. The transfer RNA will recognize that 3 nucleotide sequence called a codon on the mRNA. That might be A, U, G. Well, the anticodon on the tRNA would therefore be U, A, C. And this guy is bound onto some sort of amino acid. In this case, this would be like methionine. And then as it goes on to read the next guy, like let's just say it's C, C, C. For that codon, then this one would be, anticodon would be G, G, G. And I don't know what that codes for, but let's just say it's glycine. So this amino acid here, let's say it's glycine. And then that would link together. And the ribosome is going to keep on reading down this messenger RNA and matching up tRNAs and then the amino acids that are associated with it. And then they'll bind together to create the protein that it spits out as the product. And that is how translation occurs. Obviously, if you're making new bonds like this, bonding together amino acids to create these new proteins, you're going to need energy to drive all of this, which is the importance of our metabolism whenever we get to that. You're making new copies of your DNA, you're going to need energy for that. If you're making messenger RNA, you need energy to make the messenger RNA. You need energy to turn that messenger RNA message into the protein using your ribosomes and your transfer RNAs. You needed energy to make the ribosomes. You needed energy to make the transfer RNAs. So all this stuff is going to come full circle, because if you need to make energy to make these things, guess what else you needed energy for? You needed energy to make those freaking enzymes that are going to help you make the energy. So all of a sudden, you're constantly needing to make energy to make anything happen ever. And it really puts it in perspective, the importance of ATP. But anyways, having said all this, that's our basics for how that we did replication, making copies of your DNA. That's all that is. Transcription, making your working copy of your messenger RNA. That has to happen if I am okay. So your genes for your DNA. For eukaryotes, this happens in the nucleus. For eukaryotes, where does this happen? Just in the freaking cytoplasm. They don't have a nucleus. You can say it happens in the nucleoid region, but that's not an organelle. It's not separated from the cytoplasm. That's just part of an area of the cytoplasm. So all of transcription and translation just occurs in the cytoplasm for bacteria because they do not have a nucleus. But because we keep all of our DNA in our nucleus, if you need to get to that DNA to make your RNA copy, that has to happen in the nucleus. That's your Smithsonian institution. Bacteria are just wild and free. So they don't have anything governing that. So they can actually have transcription and translation happening at the same time. While they're making their messenger RNA, they can already start translating it because they don't have to take it out of the nucleus. We have to take ours out of the nucleus into the rough endoplasmic reticulum and then start translating it. So that's the downside of having everything compartmentalized. But at the same time, we can get more done more effectively by compartmentalizing. So that's why we're more complicated creatures. All right. Cool. We talked about the triplets. Triplets are essentially like if you had your DNA that was relating the DNA version of this version of the codon is a triplet. Okay, that's what that is. And we already talked about the importance of the amino acids coming together to build our proteins during translation. So let's talk about operons, inducible versus repressible operons. For operons, you basically have the promoter. This is going to be where RNA polymerase will bind onto in order to start transcription. So in your actual DNA, you have your promoter, you have your operator, and then you have a series of genes. Gene 1, gene 2, whatever. There might be a lot. We'll just put 2 because we don't care how many there are, but whatever. There might be more after that. And what bacteria will do is they will want to make these genes that could be associated with some sort of like lactose metabolism. They don't need to do this all the time. So what'll happen is this operon way it's controlled is at the operator, you will have a repressor. That's this blue that I'm putting here, okay? And this is on here all the time. So RNA polymerase can bind here at this promoter site, but it can't get through here. All right. So then what happens is we will get lactose to come in. It binds with the repressor to release the repressor. Okay, then that blue comes off. I'm not going to erase it because it's just too much work, but the blue then will change its shape and will no longer be on there. And now RNA polymerase can continue to make our gene products of whatever gene 1 and gene 2 are to break down lactose. We don't even do this unless lactose is there. So lactose is our thing that will relax our repressor, and that will turn on that operon. So if this is off and we need to turn it on, then it can be induced. So this is an inducible operon. Okay. For repressible operons. Repressible operons, promoter, operator, gene, we'll just say gene A this time because it's going to be different genes, gene A and gene B. These guys might be involved in making an amino acid, right? And you often need amino acids just to build things in general. So you're going to need these guys. But now your cell isn't using it up anymore. For some reason, you're just not needing to build more cells for whatever reason. And now you're going to have have an excess amino acid here. Well, as it turns out, this is essentially the equivalent of what we had above. But this is our amino acid. Okay. So we're making an excess. The cell isn't using it up. So that excess guy plus our repressor guy this is going to block our operator. So normally, if we don't have an excess of this, normal operation of things, we are having our polymerase come on here, shoot right through here, through our operator region, and we can just make our gene products A and B. Just fine. As long as we don't have excess of our amino acid product. Once we start having excess of that amino acid product, it binds onto our repressor here and changes its shape. So now it will come and block making more. This, because it was on, it's not inducible. It's already on. So we can turn this one off, which means it's repressible. That's the difference between those operons there. All right. Horizontal gene transfer. What are the different ways that we can transfer those genes horizontally? So that means that you're not inheriting it from like your parent, like the cell that you came from, that your progenitor from. You're inheriting it from cells that just are coming up to you on the street, if you will. So this horizontal gene transfer, you can have this from conjugation, which is via sex pili, which are little channels that they can transfer DNA through between one another. Transformation, we have pickup of exogenous DNA. by competent cells, right? We learned about that in lab. And then the other one is transduction. This is via phage infection. Those are the three main ways. All right, and lastly, in the genetics chapter, we talked about the mutations. This all has to do with the amino acid changes. Do nucleotide changes cause amino acid changes? Okay, so that's the question. So if you have silent mutations, you had a nucleotide change. That is a mutation, but your amino acid stayed the same. It still codes for the same amino acid. Okay. In missense mutations, you have a different amino acid with your nucleic acid change. And then in our nonsense mutations, you do not have an amino acid. You can look at it that way. It's not a true amino acid. It doesn't code for anything. And in other words, that means to stop. That's not an amino acid. It just tells it to stop because there is no amino acid code there. So those are our possible mutations. You can have back mutations. That just means reverting back to what the original code was and coding for the original amino acid. You can also have frame shift mutations where you have adding or deleting the nucleotide so that now the frame that we're reading has shifted. Like if you took out a letter in your book, but kept all of the sizes of the words the same, but just shifted the letters over by one, now the words aren't words anymore. So it's that same idea. That's what a frame shift mutation would be like. Okay. With genetic engineering in chapter 8, we talked about the size of DNA. We expressed that in terms of base pairs versus kilobase pairs versus megabase pairs. So base pairs, this is 1,000, this is 1,000,000. We talked about gel electrophoresis. DNA has a negative charge because of the phosphates. Those have a negative charge. So DNA is negative charge, so it moves to positive. Right? Also, large fragments get stuck. Smaller fragments go faster. So the smaller ones will be at the bottom of the gel. The large ones will get stuck at the top where you put the sample in. Everything's moving to the positive end. All right, so that's gel electrophoresis, using electricity to force that DNA to move throughout a gel and then separate based on size. So it separates based on size. PCR is polymerase chain reaction. This is just amplifying or copying DNA. Polymerase chain reaction. That's all it's going to be doing. And it has its basic steps of denaturation, where we're separating the DNA strands. Then annealing. Annealing is sticking our primers on. So the primers are what's going to help you find the specific gene you're trying to copy. And then we'll have the elongation step, where we're adding on our nucleotides in sequence that are complementary to the strands that you separated. Works very similarly to DNA replication. And so that's the idea of how PCR is going to work. And it just does that over and over again. You have to have the primers to look for the gene that you're looking for. So you have to know what gene you're looking for. And therefore, it doesn't just find anything. You have to know what you're looking for and have the primers for it. When we're cloning things, we have different vectors that we can use. More commonly, we're going to be using plasmids like we talked about in the lab. So you can insert DNA into those using like things like restriction enzymes, insert DNA in there, have, antibiotic resistance to help with selecting to make sure that you got the plasmid in there, and then control expression of that gene based on certain gene, ons and offs like we used with arabinose in the lab, right? So cloning vectors could be things like your plasmids. It could be artificial chromosomes that you just completely generate, or it could be things like virus genomes as well. Denaturing DNA, we typically talk about doing that with heat, but you can do that with pH as well. DNA strands are just held together by hydrogen bonds. So any way you can disrupt those weak attractions will cause denaturation of DNA as well as protein, though, right? Because a lot of our protein structure depends on those hydrogen bonds too. So keep that in mind. All right, and then restriction endonucleases, ACEs, whenever we hear that, you need to be thinking enzymes. Enzymes are typically proteins. And so these are proteins that will recognize palindromic sequences in the DNA and then make sticky end cuts typically that we can use to help insert DNA into other DNA and allow us to create genetic hybrids. Those are going to be genetic hybrids that we would call GMOs, or genetically modified organisms, and things like that. All right, chapter 9 dealt with nutrition. We talked about the CHOMPs. So this is carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur being our major components of living organisms. We talked about our nutritional and our energy sources. So if your energy is coming from the sun, then you are a phototroph. If your energy is coming from breaking chemical bonds, you're a chemotroph. So these guys are energy relating terms. Whereas auto or hetero is talking about where your carbon source comes from. Saprobes, these guys get their stuff from breaking down dead material, decaying material. Detritus is what we call that stuff. All right, our organisms that have temperature preferences are mesophiles. This is in our study sheet. We didn't talk a whole lot about it in class because we had to sort of compress this stuff, but our temperature preferences, mesophile, they like the mid temperatures. So this will be like room temperature or body temperature, stuff that we could survive in comfortably. Our thermophiles, they like the high temps, and our psychrophiles equals the low temp preference. We talked about diffusion and osmosis. Diffusion just has to deal with moving molecules from an area of high concentration to low concentration. You can even break it down even further and think of like a whole pile of bouncy balls. If you keep stacking them up on each other, they just want to keep spreading out evenly on the floor. That is also diffusion. Osmosis is just diffusion of water molecules. That's all that it is. And our membranes allow diffusion of water molecules and not other substances anywhere near as easily. So a lot of control of concentration of substances inside versus outside of the cell is maintained by moving the water molecules and not the other things that we would call solutes. So water is our solvent. It's where the solutes are dissolved in. So a lot of that is trying to keep it equal on both sides. So let's see. So if you have a cell and it has stuff in it, and you put it in a cup of water that has no stuff, obviously stuff, you can call it solute if you want, but it doesn't matter because there's no stuff in this water and there's stuff inside the cell. All of your cells have stuff in them. Let's just be clear on that, okay? So even if you don't know the exact composition, and if that mattered, I will tell you. But even if you don't, you know that all cells have some kind of stuff in them. They have to, DNA, whatever it is. And so what happens is if you put a cell in pure water, what science wants is to dilute out stuff so that it equals the same. It cannot move. We can't make this stuff go out. This cannot happen. So instead, what science would have us do, because we can't do that, we're going to move water in to try to water down the contents of the cell so that it matches the outside. It's impossible because you're still always going to have stuff on the inside, right? So what happens? The cell just keeps filling with water and filling with water until it bursts open, right? Or lyses. That's the word for bursting a cell open. So that's what's going to keep happening. This solution is called hypotonic. If the roles were reversed, if we had a water-filled balloon inside of a very, very salty solution or stuff, lots and lots of stuff in here, right? What happens now? We cannot move the stuff. We cannot put the stuff in. This doesn't work again, right? So what can you move? The water. So we're going to try to move water out of the cell to dilute out the stuff that's around it. So we're going to shrink the cell because we're losing all that water until the concentration of stuff inside of the cell equals outside. Well, if there is no stuff in the cell, it's just going to shrink to oblivion, basically. This situation is our hypertonic, right? Like when we pack in salt. And this need to give out the water is called osmotic pressure, which we get whenever we pack things in salt, when we're preserving them. So that is how those things work. We also talked about the bacterial growth curve that basically had to deal with whenever you inoculate, you have that lag phase. That's just the organisms adapting to that environment, having nutrition, making copies of their DNA. They're delayed at first. So that's the lag phase. Then you go to that log or exponential growth phase. Then you have stationary. And then you have death. These are the growth curves for bacteria. So stationary, you can imagine, this is where we don't need to be growing anymore. We've kind of reached the max limit of what your media can sustain. And then once you start losing nutrients and we have to have, cells can't live anymore because there just isn't enough nutrients available to sustain life. And we have toxic products made from cells dying. That's whenever we start seeing the death phase. So that's how the growth curve works. We also talked about symbiotic relationships. Our symbiotic relationships are going to be our mutualism, which kind of speaks for itself, right? We have both benefit. We also have commensalism, where one benefits, the other is neither harmed nor does it benefit. So nothing happens with them, but one of them's benefiting. So mutualism, commensalism, and parasitism. And you guys know this one. It's where we have the parasite harms the host. All right. Chapter 25. Again, we didn't talk a lot about this. We talked a little bit about the nutrient cycles. I am not going to ask you about the different kinds of nitrogen cycling. And I would hope that by now you know that our autotrophs are going to be making carbon compounds that the heterotrophs can use and release carbon dioxide that, again, the autotrophs will fix into the compounds. Again, that cycle works that way within those organisms, as well as including combustion outside of like our microbe cycles for the carbon cycles. So that's really our biogeochemical cycles. We didn't really talk about trophic levels, so don't worry about that stuff. Just know about how energy and nutrient movement works, generally speaking. Okay, those kind of speak for themselves. Like I said, I'm not going to ask you about like ammonification or anything. I'm not. I know it's on the study sheet. I am not asking you about it. Be aware of how water treatment plants will be working. I don't need to know step by step necessarily about these water treatment plants, but I would for sure be aware of the microbe side of things. The fact that we do add microbes in to break down things like sludge, and the fact that we use coliforms as indicators of contamination of water. So coliforms might not themselves be harmful. It might be the normal E. coli or other gut bacteria that even if you were exposed to them, you might not get sick as healthy adults. But if they're in the water, the pathogens could be there too. So if we see these guys at all, we're going to assume pathogens could get through. So coliforms are just indicator bacteria. All right, let's move on to metabolism. Chapter 10. We have catabolism, where cats break down and destroy things, just like we're going to break down our molecules to make them smaller. That releases energy. And then anabolism uses energy to build larger molecules, including muscle, like we think with anabolic steroids. So that really is, those are the two main components of metabolism. If I say you metabolize something, I'm usually talking about catabolism. So if you metabolize lactose, if you're going to break it down, then you are catabolizing it. So remember that. We talked about enzymes, their structures and their functions and everything like that. Before I get into Leo, it says Gur. Let's talk about the enzymes. We had 6 different classes of enzymes. Essentially, these were our oxidoreductases, our transferases, our isomerases, our hydrolases, our ligases, and our liases, dealing with double bonds. So those were the 6 groups of the enzymes that we had to learn about. Oxidoreductases deal with electron exchange. Transferases deal with transferring groups from one molecule to another. Isomerases just switch molecules from one shape to another. Hydrolases, we're going to be breaking bonds of things and then liasing, or I should say this, hydrolases are going to break up water to break bonds of other molecules. And ligases are going to be creating water whenever we make bonds between molecules. So remember, DNA ligase ligates or combines those Okazaki fragments or those fragments from that we're putting together from using our restriction endonucleases. Anytime you put fragments of DNA together, use ligase. That's combining and making new bonds. Hydrolases are like the opposite of that. We're breaking those bonds. And water is involved in both of those reactions. So water is either being broken or made. Hydrolases are going to be actually breaking the water, hydrolyzing something. So those are the classes of the enzymes. So what are enzymes? Enzymes are proteins. So they're folded up amino acid sequences that are actual functional proteins. The functional whole portion of a protein that works is called the holoenzyme. Okay, the whole enzyme that functions is the holoenzyme. Apoenzymes are the protein portion of the holoenzyme. Geez, sorry, I'm throwing things now. And cofactors are non-protein things that are necessary for enzymes to function. So apoenzymes ain't going to do anything if they need help from their cofactor. These are non-protein portions. They can be organic, in which they are called coenzymes. This is things like NADH and stuff like that. Those are organic coenzymes. If they are not organic, if they're like metallic things like we have with iron working with hemoglobin and stuff like that, if they're metallic, then they're just cofactors. Okay. It's just that a certain kind of cofactor that is an organic molecule is called a coenzyme. Okay. So those things will come together and work together in order to in order to create that whole functional enzyme. All right, so that's the gist of the enzymes and how like their structure is comprised. Then we have the ways that we can inhibit them. We have our competitive versus our non-competitive inhibitors. Competitive inhibitors, these guys bind at that active site. This means that our substrate that our enzyme acts on is not going to be able to bind there because our inhibitor is there instead, right? In non-competitive inhibition, we usually have some sort of regulatory molecule that will bind at an allosteric or regulatory site. So it is not the active site, somewhere else. That's the most important part, okay? This causes the enzyme to change its shape. And now our substrate can't access its active site. And that's how that non-competitive will work. So it's not competing for the active site. So competitive is literally talking about the exact active site where the reaction would be mediated. All right, let's move on to aerobic respiration and fermentation and that sort of stuff. So I'm going to clear my screen again over these notes. You can always come back and look at the recording and relook at these screens though. All right, for aerobic respiration, y'all. What's our final electron acceptor? If you cannot tell me at this point that you know that it's oxygen, then you're really going to do a lot more studying than you think you do. Our final electron acceptor is oxygen if it's aerobic. What if I told you it was anaerobic? What's your final electron acceptor for anaerobic respiration? Not oxygen. And guess what? That's all you need to worry about for this test. Okay. So if it's anaerobic respiration, your final electron acceptor is not oxygen. If it's aerobic respiration, it's oxygen. All right. So aerobic respiration and anaerobic respiration, these two things essentially follow the same 3 steps. They will go through glycolysis, then Krebs, then the electron transport chain or electron transport system. All right. Glycolysis, you are just doing glucose to pyruvate, right? Then on the outside of this, you'll take your pyruvate. So we have our 6 carbon to 2 times 3 carbons. Do you make carbon dioxide as a byproduct in glycolysis? No. Do you make any ATP? So let's do this. Let's do this. Let's do ATP. Let me erase this real quick. Oh, this is going to be smaller than this way. I want to make this make some sense, so just give me a sec. Okay, right. So ATP, or do we make our NADH or something like that? Or do we make CO2 in these guys, okay? So for glycolysis, Krebs, and electron transport chain. All right, so in glycolysis, do you make any ATP in glycolysis? Yes, you do. You make a couple. In fact, this is what powers fermentation. That's the only place that fermentation gets any ATP. It stops in glycolysis. Right. Do you make any NADH or FADH2? Yes. Do you make carbon dioxide? No. Moving on to Krebs. Now, in the Krebs cycle, you have to have gone from pyruvate and turned it into something called acetyl coenzyme A. And this is what goes into the Krebs cycle. So this happened before Krebs, but after pyruvate. So anyways, Krebs, what we're going to do is put acetyl CoA and cycle it in with some other molecules, turning it back into a 6 carbon. So it came down to 2 carbons. This was 3 carbon. This was 2 carbon. Do we make carbon dioxide here? Yes. Do we make NADH here? Yes. We don't make any ATP though. Just to be clear, what happens in that little mini step in between. So that's our first carbon dioxide emission, if you will, from all of this. So Krebs, we're going to take acetyl-CoA, our 2 carbon, recombine it with some other crap in the cycle to make a citrate and kick off some carbon dioxides. So we're basically rearranging oxygen, rearranging bonds, let's just say, to steal electrons. That's the whole point of Krebs. So our big products here are going to be our electron carriers. For glycolysis, our big product was pyruvate, so we could go into the Krebs cycle. For the Krebs cycle, our big product is NADH and FADH2. So these are our electron carriers. Okay, so are we going to make any ATP at all during the Krebs cycle? Yes. Are we going to make any carbon dioxide? Oh, heck yeah, we are. Because each step of here, while we're rearranging these bonds, we're also going to kick off carbon dioxide, so carbons during this whole step. So we'll release carbon dioxide while we're breaking down all these bonds and rearranging all these electrons to steal them off. Okay, so that's the Krebs cycle. Now we in the electron transport chain will move our electrons through cytochromes to pump H plus protons out of the membrane to create our proton gradient, which is our proton motive force. This process is called chemiosmosis. Now, we've created our proton motive force. What's the whole point of that? Protons will move back in, we'll just say through the ATP synthase. And then that will lead to the production of ATP. Right? That's the whole point of all of that, is the ATP. So of course we're going to make ATP here. Yes. We're going to make ATP here. Do we make NADH or FADH2 here? No, we're using this. What we made in the other 2 steps. Are we making carbon dioxide in the electron transport chain? No. That's a hard no. A lot of you guys think because we're using oxygen as our final electron acceptor in the electron transport chain, that must mean we're making carbon dioxide, but you're still confused. We never make carbon dioxide in the electron transport chain. We only make carbon dioxide in the pre and the actual Krebs cycle. It's the only time that you make carbon dioxide as waste. Okay. So what is the whole point of oxygen in the end of all of this? Well, after you've moved all of these protons here, you want to keep creating this proton force. You can't have them move in and set up shop. You also can't keep having these electrons move through and stay at the end. Your last cytochrome is cytochrome C oxidase. It will take that last electron and move it to oxygen along with your protons to make water as your byproduct. So you use your oxygen to accept those electrons and the protons. We have water as our byproduct. And now we can reset this whole thing, maintain our gradient, and move our electrons through to keep making ATP. And that's how aerobic respiration works. Anaerobic respiration works the exact same way. We just don't use oxygen at the end. We use something else. Fermentation only goes through glycolysis. It never goes through the other steps. All right, so photosynthesis. In photosynthesis, you have two cycles. You have the light-dependent reactions and the light-independent. This is also known as the Calvin cycle. There will be questions on the test relating to the Calvin cycle. I don't need you to know that it's even called the Calvin cycle. As long as you know that the things that I'm asking you about are not called the Calvin cycle, you'll be fine. As long as you know things are called glycolysis, Krebs, electron transport chain, then you will know that Calvin cycle is not part of aerobic respiration, is it? No, it's part of photosynthesis. So these are the two steps of photosynthesis, all right? The light-dependent reactions, you just have photons hitting basically what are the equivalent of cytochromes, okay? But they're pigment molecules. So those pigments get excited, they release electrons, we create a proton gradient. ATP is made from ATP synthase. That ATP then can be used in the dark reactions or the Calvin cycle or the light-independent reactions. It's all the same thing. So in the Calvin cycle, we will use that ATP and carbon dioxide from our environment and fix it into organic compounds like glucose. We can then use glucose to make energy in aerobic respiration. Oh my God, let's not all faint all at once. But yes, plants have mitochondria and they have chloroplasts. In the chloroplasts, they make the ATP that they will use to fix into glucose. And they'll move that glucose over to the mitochondria and they'll get buku ATP out of that glucose. So a lot more ATP. So now we can actually build things in the plants and make new cells and things like that. So that's the whole point of photosynthesis. And the light reactions making the ATP, the dark reactions or Calvin cycle making the glucose, and then shooting that off into aerobic respiration. Y'all, you have any questions about it before we move on? Chapter 13, we talked about infection and disease. This was a pretty straightforward one. This is where we start really pressing home that idea of that microbial antagonism, where you take up the stadium seeds, you're going to make antimicrobial peptides, you're going to use up the nutrients so the bad guys can't have them. That's what our microbes are going to do for us in our microbiome. When you infect somebody, you got to get in, you got to stick there, you got to survive being destroyed by the host defenses. Then you get to cause your disease by making your toxins or breaking down the tissues or whatever it is you do. And then you got to exit to go infect the next guy. So those are the basic steps of infection. In order to survive your host defenses, a lot of these guys have to have some way to avoid phagocytosis. Either you live inside of the phagocytes, or you kill the phagocytes, or you avoid being phagocytosed altogether. That often will, the capsules will come into play there. Once you start causing disease, any aspect of an organism that allows it to create pathogenesis and therefore cause disease and set up shop and really grow and cause infection, anything that helps with that is a virulence factor. That's the term for that. This often includes things like exotoxins that they excrete. Even endotoxins can be a virulence factor because it helps aid in the inflammatory response, and that inflammatory response can actually help out with the pathogenesis or the disease causing aspect of the organism. When we start looking at infection and disease, we can start looking at signs and symptoms that are produced as part of the disease. If you don't have signs or symptoms, you don't have disease. So our signs are going to be measurable aspects versus symptoms being non-measurable. Measurable might be the increased temperature of a fever. It might be increased white blood cell count and things like that. Whereas symptoms would be just reportable things like pain. That's a big one, or a fatigue. You can't measure that. All you can do is go by what the patient is saying. All right. So if we're talking about inflammation, there's certain aspects of inflammation that can be measurable, right? We can look at your inflammatory cells that are involved in the response. We can talk about the steps of inflammation as well. We can talk about, did I not even include that in here? The calor and all this. Oh, I can talk about that in the inflammatory response stuff. Yeah, looking at different aspects of inflammation, we'll talk about that in chapter 15's review. We're getting there though, y'all. All right. And then measuring like leukocytosis or penia, having too many or too few white blood cells is an actual measurable sign. Septicemia and bacteremia are also signs. You can actually diagnose that physically. And then our asymptomatic versus subclinical infections, latency and sequelae, these can be a lot harder to diagnose because with our latency and our subclinical stuff, because you're not having any signs or symptoms to even know that you're sick, how would you even go get diagnosed? And then sequelae is a term that's referring to lasting effects of an infection that can be permanent in some cases, but you've already cleared the infection and you're still having these effects of it. All right, there's 4 phases to our infectious disease process, and they actually coincide pretty well with our growth curve that we talked about with our bacterial growth curve. Incubation, that pretty closely mirrors our lag phase. So the organism is just getting introduced into the body and hasn't started eliciting an immune response. It hasn't started really truly multiplying. It's just getting oriented and finding its place in the body. The prodrome is whenever you have your first signs or symptoms apparent, the very first ones. You might just start with scratchy throat, for example. Then you will have the acute phase, which is also known as the invasion phase, where you have the worst of the symptoms. So you have your full-blown expression of the disease. And then the convalescence phase. Convalescence is referring to the clearing of the infection and the reduction of the symptoms. Now, you could go into latency here and not clear the whole infection entirely, though. All right, reservoirs are places where these diseases exist just naturally in nature. They don't necessarily even cause diseases in these organisms that are, or places that are reservoirs, but that's just where they're maintained in nature. The source is the place where you got sick from whatever you are sick with. That's the exact thing that you got sick from, not all reservoirs altogether, but the exact place that you got sick from. The carrier, these are people that are infected that are not true reservoirs. They are infected, but they are carrying the disease around without signs or symptoms, typically for whatever reason. They could be recovering from the disease or newly infected, but still contagious and haven't had symptoms yet. Or it could be that the organism is hiding out in part of their body that aren't causing signs or symptoms, but they can still be transmitted. Zoonosis is any sort of infection that comes from the animal population, primarily, like rabies. All right, transmission terms that we should be aware of. Communicable means it can be given from person to person, whereas contagious means very communicable. It's just another level of it. Non-communicable, these are diseases that you can't get from another person, such as like type 2 diabetes, for example. Ways that things can be transmitted. We talked about vehicles, things that are able to allow transmission of a disease, and fomites are like actual stationary, non-living objects that, like, if you borrow my phone after I have had COVID and you put it up to your ear and you get my germs from my COVID on you and now you're infected, that's a fomite. My phone was a fomite. Is my phone infectious? Is it an actual reservoir? No, it's just a vehicle for transmitting those microbes at that moment. So it's a fomite. We also have oral fecal transmission, water soil transmission, and air and droplet nuclei versus aerosols, just transmitting things by coughing or sneezing in the air. And those things can become dried and last longer in the air, or they can just be transmitted directly from the water droplets that are sneezed, whatever. For epidemiology stuff, all right, we already know about Florence Nightingale and John Snow. Remember, Florence Nightingale helped out in the Crimean War. She figured out that people need to be basically practicing hygiene. John Snow, she was the original nurse. So she's the mother of nursing, basically. John Snow is the guy that figured out there was a cholera epidemic and that he traced it down to this particular pump in the Soho district of London. And he removed the handle from that pump, and then people couldn't, drink that water anymore. And he saved everybody from cholera. So he's the father of epidemiology. All right, our terms: prevalence versus incidence. Prevalence is talking about the overall total number of infections in the outbreak, versus incidence is how many we've had in the last week, how many we've had in the last month in this particular area. So it's usually a chunk of time, how many cases we've had in a chunk of time, whereas prevalence is overall up to now from the beginning. Morbidity rate is talking about incidence. So the morbidity rate is talking about how many cases we've had in the last week, for example, or month, whatever. Mortality rate is talking about how many deaths we're having with this outbreak. Endemic are diseases that occur naturally in that area. Nobody's going to be too surprised if you come out with a cold, right? This is an endemic type of a thing. Whereas epidemic, now we have an outbreak that is exceeding what our expected, outbreak numbers would be. It's exceeding the prevalence. So do you usually have Ebola in your, area where you live right now? Probably not. So if you have any cases of Ebola, now we're exceeding into that epidemic. It's exceeding that number of cases than what's expected. Now you can have sporadic outbreaks. So if you had somebody in your area, like a person that got Ebola, that's not an epidemic. That is a sporadic outbreak that person got from visiting, Africa or something and contracting that disease. Sporadic outbreak of rabies. The kid down the block got bit by a rabid raccoon while he poking it with a stick. And now that kid has rabies. That's a sporadic. That's not an epidemic, right? But once we have numbers of people that are actually being affected in the population, and it seems to be spreading around outside of the numbers, what you expect in that area, that's an epidemic. A pandemic is whenever an epidemic spreads across continental divide. All right, studies that we use to investigate these things, we have cross-sectional. So you're looking at factors of the disease at a specific cross-section in time. So you're looking at this specific point in time, how many people have this disease and what symptoms they have or don't have, for example. So this is cross-section, taking a cross-section of that specific moment in time. In case control studies, we are looking at people who have the disease versus the control group. And then looking at the differences between the people that have the disease and the people that don't have the disease right now. Whereas the cohort studies, we are following people that are in a group that don't have the disease yet and looking to see if they get the disease. Now, that is an example would be people who smoke versus people who don't smoke. None of these people have the disease. We'll get a whole cohort of people that smoke and a cohort of people that don't smoke. Nobody has cancer yet, but we're going to see who does. That's a cohort study. Nobody has it yet. We're going to follow it over time. Case control, you have cases that have it and people that don't, and you're comparing them. Cross-sectional, you are looking at that moment in time. Do they have the disease or not? Do they have the symptoms or not? That sort of thing at that moment. Okay. R naught is the measure of how many people you're going to infect when you have a disease. You get COVID. How many people are you likely to infect? It's between 3 and 5 people, whether you have symptoms or not. So that is the R naught for that. CFR is case fatality rate. That is how many people will die of that disease if they don't have any treatments or vaccines or anything. So the likelihood that you will die of rabies if I don't treat you is the case fatality rate is 100%. Same thing with HIV. Case fatality rate for HIV is 100% if you're not getting any treatments, right? All right, let's move on to Unit 3. Microbial control mechanisms. We've talked about physical, chemical, and mechanical aspects. I would be able to at least categorize each control method as one of those. So obviously, if we're using a chemical like bleach to clean something, that's the chemical. And that's pretty straightforward. Mechanical, I usually don't separate it from the physical. So I would really just look at this as physical or chemical. Mechanical, here we're actually removing any microbe from the situation, not necessarily killing it, So mechanical removal would be like scrubbing a microbe off of your skin or filtering it out. So you're actually removing the microbes from those situations. Those are mechanical. So that's the difference from physical, where you might just be killing them where they are on the surface, like we do with UV radiation or something. But that's the difference there. So really think about these as being more like physical versus chemical. And if you want to subdivide the physical down into mechanical, that would help with understanding of the difference between what mechanical might mean versus just physical. All right, so for our terminology for getting rid of our microbes, we have sterilization, where we have elimination of every living thing. We have disinfection, where you're removing most things. Usually what's going to survive is the endospores. Antisepsis, where we have application on living tissue. And then decontamination or sanitization, where we're now talking about mechanical removal of microbes in general. All right, like scrubbing and things like that. Jeez. All right. Let's talk about microbicidal versus microbistatic. Again, basics for you guys. You guys know this one. Cidal, we're killing. Static, it's not being killed. We're just stopping it from growing. All right, so our microbial control mechanisms can target certain aspects of cells, whether you are physical or chemical, it doesn't matter. You're going to be targeting the cell wall, membrane, the nucleic acid synthesis or function of nucleic acids, or you're going to be denaturing proteins. And that's the general gist of how we would be working. You'll want to know which things are going to be able to sterilize versus which agents are going to be able to disinfect. And a lot of the stuff you guys actually probably know better than you think you do, honestly. It's just the chemicals, I think, that are going to trip you up more than anything, knowing which chemicals can sanitize versus disinfect. So make your flashcards for that stuff. I do have the chart here to remind us about this. But again, this chart is probably your gold as far as making flashcards on what can cause disinfection and what can cause sterilization for our physical agents. For our chemical agents, I would go back to the study sheet and like really break it down as far as what can do what. For example, our halogens. Halogens, which includes our chlorine, iodine, and bromine and things like that, they can sterilize when used appropriately. Aldehydes can sterilize if we're talking about glutaraldehyde, for example. Formaldehyde, not quite as much. And the OPA one, orthophthaldehyde, that one doesn't sterilize, but it's safer to use. So you might want to brush up on those types of things. I'm not going to go through the list of those because that really is more like a list of things that you guys can make your own list of and remind yourself of that. I'm going to go over the concepts. I'm not going to ask you how those agents work, but I might ask you, like, you should know that incineration uses dry heat. Okay, be aware of that. But as far as how our chemicals work, I'm not going to ask you if the alcohols affect a cell membrane or cell wall. I'm not going to get that in depth on our chemicals, even if it might be on the study sheet. I just want you to know which ones can sterilize and which ones can disinfect and which ones might be useful for, like, you might want to know you don't use ethylene oxide, that gas that explodes. You don't use that to like prep skin, for example. So have general understanding of our chemicals and whether they can sterilize and whether they can't. I'm not going to really go beyond it on that on the final exam. Okay. For the physical agents, those should be kind of self-explanatory based on how you're categorizing them on how they're going to be working. Keep in mind that the heat generally targets the destabilization of the proteins. So we're really like denaturing proteins and DNA usually also at the same time with heat. So that's what's really going to be causing the problems. And if you are sterilizing, we're usually completely destroying total protein structure with the heat. Where did my pen go? There it is. Okay. So that's what I'm going to say about these microbial control mechanisms. I'm not going to go over each one. For our drugs, we know the Kirby-Bauer assay is used to test for drug susceptibility. The bigger the clearing, the more susceptible you are. And then you can use that information to determine the minimum inhibitory concentration needed to treat an infection based on how big that zone of inhibition is, right? That can give you an idea of how to select our correct doses. And you also want to consider the ratio of the amount of drug that will cause toxic problems versus the amount of drug that it takes to actually be effective. That's our therapeutic index. So we're comparing if it takes 50 milligrams of this drug in order to be toxic, but it takes 50 milligrams to actually be effective to cure the illness, that's a problem. That's a 1-to-1 ratio. And that could be causing actual toxic problems that we might want to avoid, and maybe choose a different drug that doesn't have as much of a risk of toxic side effects. That's the importance of the therapeutic index. And it also brings into call that idea of that selective toxicity. We want to be as toxic as possible to the bad guy, the pathogen, without causing any harm to the host, if possible. That's selective toxicity. No toxic to the patient, fully toxic to the pathogen. All right, so our antibiotics also target cell wall, cell membrane, nucleic acids, and proteins, just like our control mechanisms. We also hacked on another group area, which is our folic acid inhibitors. So our cell wall ones that we want to be aware of are our arcillins, which are beta-lactam drugs. They work on inhibiting cell wall synthesis. Then we have carbapenems and cephalosporins that fall into that category as well. Our cell membrane inhibitors are going to be their polymyxins. Our nucleic acid inhibitor, our synthesis inhibitors are going to be our fluoroquinolones. They end in the word floxacin. Our protein inhibiting, synthesis inhibiting guys, they inhibit the ribosome function. These are aminoglycosides. A lot of these end in mycins. A lot of our other group, the macrolides, also can end in mycins. So a lot of the mycins are going to be our protein inhibitors, right? We have tetracyclines and glycylcyclines. These cyclin guys are derivatives of the aminoglycosides. They still are protein synthesis inhibitors. We have our folic acid synthesis inhibitors. These guys are usually competitive or non-competitive inhibitors along different enzymes in the folic acid synthesis pathways in bacteria. These are generally our sulfa drugs. So we have sulfa methoxazole, and then we have trimethoprim that has meth in it. So it either has meth or sulf, and those are like methane is stinky, sulfur is stinky. And I think of those as being like the stinky, like sulfur type drugs. And those are dealing with our folic acid synthesis inhibitor pathways. Then we have our antifungals. These are usually going to end in azole or be amphotericin B is another one. So amphotericin B or like miconazole, fluconazole, ketoconazole, they all end in azole. There's only one azole that doesn't belong in the antifungal group, and that is metronidazole. And I remember the difference because the metronidazole went to the big metro to be part of the big city to go deal with the bigger, guys. So that would be our protozoa as our amoeba fighting guy. So it's anti-amoebic. That's what the metronidazole is. Metronidazole also is good for anaerobic. That's what I'm trying to say. Anaerobic infections. So it will be treating that as well for bacteria. So it's not antifungal though. The metronidazole. It's the only azole that went out to the big city and did other things. Quinine is for, and its derivatives like chloroquine, anything quin. Those guys are for malaria. And then we have our antihelminthics, which are mostly we talk about praziquantel and ivermectin. There are other ones out there, but those are the ones we talk about most in the class. Our antivirals that we need to learn about were like Tamiflu and Relenza. Both of those guys are going to affect the neuraminidase function of flu, which we learned about in our flu talk. Does break down mucus and helps with entry and exit of the virus during the viral life cycle. So we can stop that enzyme from working on the surface of the flu. Then that helps with preventing infection. And we have acyclovir that helps with preventing nucleic acid synthesis in a lot of viruses, but it's used primarily for like herpes viral infections, but you can use it for other virus infections as well. All right, into our innate immune response, our first line of defenses, don't forget our chemical and physical barriers and things like keratin, our waterproofing protein of the skin, and lysozyme that breaks down peptidoglycan. In the immune system, once we get into our actual immune system response, we have to be able to recognize self versus non-self based on surface markers on the skin. And or not the skin, on the surface of the cells. It could be skin cells, any cells at all. So there's various markers all over our cells that we can recognize as just self But one of the biggest ones that we learn about in chapter 16 is our MHC, right? So our major histocompatibility complex, especially MHC1, which is present on all nucleated cells, and is our way to raise a red flag saying that we got infected, but its presence there is also a sign that it is self cells. So surface markers like that tell us the difference between self and non-self. Our lymph system consists of our lymph vessels, our lymph nodes, our lymph fluid, which is just fluid coming from the extremities to get cycled back into the circulatory system back towards the heart. That's all pumped by the muscular system. And it gets cycled through and filtered through the lymph nodes where a lot of our white cells will like kind of hang out and screen the guys that come through. The spleen is part of our lymphatic system as well, filtering out red blood cells that have been damaged, but also housing macrophages that can help screen the blood itself. White blood cells are called leukocytes. All white blood cells are called leukocytes. You can break it down into two major groups, the granulocytes and the agranulocytes. Granulocytes contain granules that are involved in the inflammatory response. Those granules are present in the phils, so that's all of the phils, neutrophils, basophils, eosinophils. Those are all granulocytes. They have granules. There's one other kind of cell that has granules called mast cells, which we only really talked about in chapter 17. Then we have our agranulocytes. Our agranulocytes are our monocytes that become dendritic cells or macrophages whenever they move into the tissues, and our lymphocytes, which are our T cells and our B cells. And that's really all you need to know about the granulocyte versus agranulocyte kind of distinction. Our granulocytes are, directly involved in the innate immune response. These guys, especially our neutrophils, they're going to be our phagocytes that are going to go out and eat up the bad guys that they recognize as having PAMPs. And those are pathogen-associated molecular patterns. Some examples of PAMPs would be LPS, so lipopolysaccharide, aka endotoxin on gram-negative cells. Or it could be like double-stranded RNA that we don't have in our bodies, but viruses can have as their genome. And peptidoglycan, like we should not have that in our bloodstream. So if our cells were to see that hanging out, we would be like, PAMP, obviously this is a bad guy stuff. So any obvious markers like that, they don't provide any memory. They just trigger our inflammatory response, just generally. We see bad guy is here. We don't know what exact bad guy it is. We don't care, but we're going to start our inflammatory response. Your monocytes and your macrophages and your dendritic cells have a part to play in both sides, the innate and the acquired immunity. They're going to clean up what's going on with the innate response and use the parts of what they pick up from that to present it to activate our acquired immunity, which we'll talk about next. Phagocytosis is one of the ways that our white blood cells are going to go around eating up all like the junk, the bad guys, the dead cells, the debris, whatever it is. So phagocytosis is cell eating, and this involves chemotaxis, which are getting called to the area where we need to eat things up by chemical signals. Those are our cytokines that do that. So we're responding to cytokines, getting called to areas and getting activated to act certain ways. We have to adhere to the microbes and to the surfaces in the area so we can start actually activating our responses appropriately in the area where we have the inflammation. Adhering to the bad guy that we're going to eat up, usually by recognizing a PAMP on their surface, engulfing them into a phagolysosome. A phagosome would be like the original. You eat them up into a vesicle that's called a phagosome. It merges with a lysosome that contains toxins in it that are going to kill the bad guy. And when it merges, it's called the phagolysosome. We destroy and we kill the bad guy in that phagolysosome. It takes about 30 minutes. And then we release the debris. When we release the debris, if we are neutrophils, we just poop it out. Macrophages will come and clean that up and then wave around anything that they find from the outside on their MHC2 exogenous flag raising, right? Raising the flag saying, I found this that the neutrophil left behind. Help me activate the correct pathway to start the acquired immunity, which we'll talk about next. In the inflammatory response with our innate immune response, we start making cytokines that will help stimulate the following. Our rubor, which is the redness in the area, we have increased blood flow. That's what causes the red. The calor, where we have heat emitted, again, usually from blood flow to the area. The tumor, which is the swelling from the fluid into the area, the cells move in to attack. They come in with the fluid and the cytokines and the nutrients that they need in order to perform their job. The dolor, which is the pain that's associated with the damaged tissues and letting you know that there's something going wrong with your body. All of these are important for helping stimulate the immune response. The heat, in fact, helps increase our metabolism and the activity of cells in those areas. It also stimulates even more communication between our cells and help with the healing process and with killing off any bacteria that are in the area that are sensitive to heat. Again, that's what fever will do for us as well. Fever will raise our metabolism. It will inhibit temperature-sensitive microbes from growing, inhibit their enzymes from functioning, but help our enzymes function better. It will help stimulate our acquired immune response and all that. So anything that elicits fever is called a pyrogen, and endotoxin is one of the main pyrogens that we know can stimulate fever from our inflammatory reaction. Interferon is a molecule that is made by infected cells to let nearby cells know that they're infected. So especially with viral infections, it's their way to signal their neighbors that they're being infected so that the neighbor cells can start making antiviral mechanisms to prevent infection. The complement cascade, we have a whole slew of about 50 proteins that are just always present in the bloodstream that don't need to be necessarily manufactured. They're just present. They're just waiting to be triggered. And it's a cascade effect. You trigger this one protein, it'll trigger a couple. Those couple will trigger yet each of them even more and more and more and more. So it's the complement cascade. So initiation gets triggered by some sort of initiating factor. There's three things that can initiate complement cascade. Antibody, which is the classical way. Lipopolysaccharide and similar repeating things like LPS, that's our alternative pathway. And then we have lectin pathway, where we bind mannose, which are sugars on the, or similar other sugars on the surface of like bacteria and stuff that we can automatically recognize as bad guys, so PAMP. So those specific things can trigger the initiation. Then we have amplification of that whole cascade. So you trigger one part of the complement pathway, and they'll trigger, several. And then the one below that triggers several itself. And you have multiple of them doing that. So it's amplification of that signal, essentially. Then we have polymerization of the products. They come together to perform their functions. And that can include the membrane attack complex, which pokes holes. That those proteins will come together, those complement proteins, once they've been activated, come together to poke holes in the target pathogens. And then they'll release their contents, and then toxins can get in there to help break them down from our immune response and everything like that. So that's a useful thing that just proteins in your blood can just do at the drop of a hat. They can also help with opsonization. Opsonization, again, is a way of helping out phagocytosis. It gives our phagocytes a handle to grab onto on things that are otherwise hard to grab onto, and can also lead to inflammation, activation of the inflammation response. When I say cytolysis down there, that is cytolysis. That's breaking open the cells from that membrane attack complex being, bursting open those cells. For our specific immune pathways, we have our B cell receptors and our T cell receptors. Keep in mind that those B cell and T cell receptors are what recognize the antigen. So an antigen is anything that your body can respond to as foreign. And antigens in specific immunity or acquired immunity, this is very specific to that one receptor. So that T cell receptor recognizes this part of the spike of COVID. And that's what it recognizes. It recognizes nothing else. So these B cell receptors and T cell receptors are what are actually recognizing that part of that antigen, the epitope, right? That's the word for that part that we're recognizing. So they recognize that. MHC and MHC1 and 2, those guys, they don't discriminate. They will present anything they find. There's not an MHC1 that is specific for the T cell receptor, or like I say, I'm not really wording that right. There is not an MHC1 that is specific for the spike protein of that part of the spike protein of COVID. MHC1 will show spike protein of COVID. It'll show parts of tuberculosis in your cell. It'll show broken down little bits of cancerous things that are going wrong in your cell. It'll show whatever the frick is inside of your cell that doesn't belong there. It'll pop it out there. Whereas a T cell receptor, that specific T cell receptor recognizes only that part of the spike protein or whatever. So that's the idea with how those are going to work. So those B cell and T cell receptors are extremely specific to that epitope on that antigen. Okay. So we have all these different kinds of B cells or T cells, our lymphocytes, that could potentially recognize all these different antigens. Well, when our cells were making up all these guys, we had to get rid of the ones that saw self, and we had to get rid of ones that were very weakly responding. So that's the clonal selection process. We get rid of anti-self, and then once we are activated, we activate only the strong responders to that specific antigen. We don't activate our entire repertoire of everything ever. Only the ones we need. We can only activate our specific immunity using our antigen presenting cells. So especially like if we're talking about MHC2, MHC2 is only found on our antigen presenting cells. These guys pick up outside stuff. So the pooped out waste of the neutrophils. We can even eat up our own bad guys and say we found these guys outside, and this is what we found from outside on our MHC2. So that's our macrophages, our monocytes and dendritic cells. Those guys, again, macrophages and dendritic cells come from the monocyte moving into tissues. And we can also include our B cells. These are the only ones that have MHC2 and can therefore trigger activating our T helper response. T helpers are the ones activated by MHC2. Okay. MHC1 interacts with our cytotoxic T cells, and we'll come to that in a minute. So what happens is you have our antigen presenting cells. They find bad stuff. They present it that are outside of the cell. They present it on their MHC2. They go find the matching T helper cell. The T helper cell's T cell receptor sees the antigen specifically held by that MHC2 and says, I'm receiving this from self and you are bad guy. So now I'm activating myself and telling you to do whatever your duties are as well. So now the T helper cell is activated and it can produce cytokines that can favor the response necessary for whatever type of antigen, whatever signals it's getting from the presenting cell. So we have our T helper cell. Now it is going to recognize antigen plus MHC2. It is not interacting with MHC1 ever. Okay. So our T helper cells can become TH1, TH2, or regulatory or memory. For our TH1 cells mostly are going to be activating macrophages and our CD8 positive cells. So when a CD8 positive cell goes on with its CD8, recognizes MHC1 that is waving a red flag saying, help me, I'm infected with a virus, with its MHC1 flag, the T cell receptor on that CD8 positive cell sees that antigen. The CD8 recognizes it as self asking for help, but it can do nothing without the signal of the TH1 cell. That TH1 cell was activated by antigen and MHC2, right? So those are separate from one another, but they need help from one another in order to activate that response. Because that CD8 cell is then going to get activated, it can now go out and kill self cells without the help from the TH1 cell anymore once it gets activated. We don't want to trigger that unless we are for sure wanting to go out and kill our self cells that are infected. Can't let that go out of control. Our TH2 cells, though, they help with activating our B cell response. So they'll go out there and help with the B cells to differentiate them into plasma cells and memory cells and regulator cells of their own and trigger the antibody response. So as far as antibodies go, B cells that are activated are called plasma cells to make antibody. Those are our effector B cells. And the antibody is just proteins that consist of two regions, the FAB, which is our variable region that recognizes the antigen itself, and the FC, which is the constant region, which actually determines the function of the antibody and determines its class. So this is whether it's going to be IgM, IgG, IgA, IgE, or IgD. So those are the 5 classes. IgG is our workhorse. It does most of the work, but we don't get it right away. The one we get first is IgM. So while we're still maturing and switching classes of antibodies based on signals we're getting from our nearby cells and all of that, we have to make IgM first. IgG has more that it can do for the body and will make a ton more of it, and it's more effective, but it takes a while to switch to that class. IgA is our secretory mucosal antibody that works better for the mucosa. The IgG doesn't work as well in mucosa. It really just works in the bloodstream. IgD is our B cell receptor. That's what it is. And then IgE is associated typically with like parasitic infections or allergies. So those are the 6 different versions of those. For our antibody functions, what our antibody actually does for us has a lot of functions. It can cause agglutination. So we'll have the pathogens or their parts sticking together so they can't do their functions, stuck together by those antibodies. We can cause coating. So we can actually like literally just coat specific coat the whole surface of a pathogen so it can't interact with what it's supposed to. We can neutralize specific parts of the pathogen, like the spike protein specifically, so it can't interact with its target to open the key, use the key to unlock the lock of the cell to get inside. Well, if we have antibodies that are blocking that interaction, that's the neutralization aspect. We can block toxins that are produced by the bacteria. So we can bind with the toxins so they can't do their function, like we have with our protection from our tetanus vaccines. And we can also have opsonization. So the antibodies can stick on to those capsules or other things that can make it easier for our phagocytes to grab onto them and eat them up. So those are the functions of our antibody. We can also activate the complement pathway. The complement, again, we talked about that, but it has its own functions. It helps with opsonization. It helps activate the inflammation response, and it creates that membrane attack complex to poke holes in. So that's what our antibody does. When you're creating your immune response, you can have different versions of how you react to things. You might be naturally exposed to something and create your own immunity against it, like you would if you actually had the chickenpox, and then you'd be immune to it. That's natural active. So natural being you exposed to it naturally, active being you created your own immune response against it, and now you have memory. If it's passive, though, the passive means that you were provided antibodies from an outside source and you're protected while those antibodies are there, but you're not making those antibodies and you don't have memory. So that's passive. If you did that naturally, we see that in the case of like breastfeeding. If you did that passively, then that's like getting an immune globulin infusion or using Humira or something like that that has a monoclonal antibody that has a function in protecting against the inflammatory response or whatever. Whereas artificial active, that artificial active means we are exposing you to something artificially, like from a vaccine. You didn't encounter it naturally, but you're still creating your own immunity against it actively. We talked about the different kinds of vaccines. I'm not really going to get into them too much. Just know that there are different versions that might be just in the skin, under the skin, in the muscle, and delivered if they were killed versus live, attenuated, or just parts of the microbes as well. It just depends on what elicits the best response. We can use adjuvants to try to help initiate our response as well. Herd immunity refers to how many people need to be vaccinated from a disease to protect the rest of the population. And that often has a lot to do with the R naught and the contagiousness of the disease. All right, cranking on to our immune disorders. We talked about 4 different hypersensitivities. Type 1 hypersensitivity was our allergy related. This was mediated by IgE with our mast cells. The mast cells will wear the IgE like little flags. And when mast plus IgE recognizes the antigen, cat dander, let's say, it releases the histamines and things like that that lead to presentation of allergy. You can have allergy that is atopic, so it'd be local, or you can have systemic, which is anaphylactic shock. You will better to treat anaphylactic shock with things like epinephrine. That is temporary, but it will allow you to dissipate those symptoms in time to get you to an emergency room to get proper treatment for that inflammatory reaction that would be happening. Type 2 hypersensitivities, these are our blood hypersensitivities. So you're either targeting ABO groups or you're targeting the RH group. Remember, if you have a type A, to you, B looks foreign. If you have a type B blood type, to you, A looks foreign. If you have type O, everybody has that as their base. Still think of O as like the original. Okay, it's like the OG. Everybody has that. and then A is like an extra bit, or B is an extra bit. You can have just A as your extra bit, but you're going to see B as weird. If you have B as your extra bit, you're going to see A as weird. If you have both A and B, you don't see B as weird. You don't see A as weird, and you don't see O as weird. Everything's kosher. So if you're O, you can only recognize O. A is weird to you, and B is weird to you, right? So think of those as reasons why you might attack the other blood groups. You're literally going to have antibody attacking those other blood groups and creating a complement cascade that's going to lyse those. Your body will then see that lysis as basically this horrible thing going on in your body and create a shock response where you'll have hypotension that can be linked to hypoxia. You'll have anemia from breaking open these red blood cells that you're attacking, and all of these things that can lead to shock and eventually death as a result of having the wrong blood type given to you. RH is really important as far as like mothers and their babies. We don't see as much severe hemolytic anemia occurring from ABO mismatches in mothers, but we see it quite strong with RH groups. So that's what we focus on when we talk about hemolytic disease of the newborn. That is going to be the RH dependent one. If you are a mother who is RH negative, your body will make antibodies against RH positive baby, right? And the first time that happens, it's just a sensitizing dose. The second time that you see it, now you have antibodies against it, just like you would if you'd had been vaccinated against RH. Your body sees RH as foreign, you have antibodies against it, and now your antibodies are going to attack it immediately. And so that can lead to death of the baby and very severe health consequences if the baby were to survive. You can treat this by giving our ROGAM to prevent the antibodies from attacking the baby. Essentially, if they're there or not, we can prevent them from attacking the baby, and that's what ROGAM does. If you're RH positive mother and your baby's RH negative, you don't see that as weird. You don't make antibodies against it. So if you're an RH positive mother, you can have RH negative babies, RH positive babies, you're not at risk. All right, type 3, immune complex mediated hypersensitivities. Here we have just antigen antibody complexes that get stuck in basement membranes of the epithelium, and we have the inflammatory response occurring to those. So like neutrophils going out there and seeing these antibody antigen things that just got stuck after having an infection and causing the inflammatory response in them. We had our specific example was post-streptococcal glomerulonephritis, acute post-streptococcal glomerulonephritis. That's what it was. And this is getting those complexes stuck in the basement membranes of the kidney while we're trying to filter things out. And then the neutrophils see that stuck there and they start attacking it, and that causes inflammation and damage of the kidney. So that was our example for type 3. Type 4 deals with the T cell response, which is why it's delayed. Even our memory T cells, we don't just have like all of them ready to go at all times and they're just waiting. We have memory ones left, and we have to reactivate those and then multiply those back out. So it takes some time for the memory response to respond with our T cells, which is why it's delayed. So this is cell mediated. We have our T cells actually responding. And in graft rejection, a lot of this is mediated by our CD8 cytotoxic T cells responding to something foreign, seeing MHC1 on, your donated kidney, and seeing that as MHC1 asking for help with a red flag, holding up an antigen, even though it's not. It just looks slightly different than what it's used to. So that's what graft rejection is based off of. You can have rejecting a graft of tissue like your body responding to that graft's MHC1 and seeing it's foreign and attacking it. Or if you get lymphocytes from bone marrow donation, those lymphocytes and other cells from your bone marrow will have their own their own T cell receptors that'll see your MHC as foreign and attack your whole body. So that would be graft versus host rejection, which is another thing that can happen. It's like not as obvious or as common as the host attacking the graft, like what we see with the kidney, but it can happen. And there's different kinds of grafts you can get. If we're talking about getting a self graft, that would be an autograft. So it's just tissue from another part of your body. Isografts are grafts from an identical twin. Then you have allografts from other people. and they're not your identical twin or yourself, anybody else. And then we have xenografts from animals, like you're getting a heart valve from a cow or something. A lot of autoimmune diseases are based off of forbidden clones that didn't get deactivated. So clones that see self as foreign. They just didn't get deactivated in our clonal selection process for whatever reason. The wrong signals were there at the wrong time. And molecular mimicry, where a lot of our pathogens, some of them anyways, like strep, can have molecules on their surface that look like our molecules. And we try to attack strep and we end up attacking ourselves because it looks so similar. And so autoimmune diseases often arise from that. That's called molecular mimicry. And immunodeficiencies, a lot of the deficiencies in our immune system, what we call hyposensitivities, they'll either be primary or secondary. Primary immunodeficiencies are going to be, gosh, dogs, you can hear them all the way across the house. Primary is going to be anything that's present from birth. And so like genetic defects that are leading to issues with your immune system. Secondary immunodeficiencies, these guys are going to be things that you acquire. So you got sick from something and it caused your immune system not to work well, like what we see with HIV, obviously attacks the immune system, and your immune system doesn't work as well because you acquired that after birth. Getting cancer, you might have the cancer itself making your immune system weaker, or you could have the treatment for your cancer causing your immune system not to work as well, like radiation is going to kill off basically your white cells too. So that's also an acquired way of having an immunodeficiency. Finally, we have chapter 18. This is our diagnostic chapter. Remember, we have 3 types of diagnostic tests. You had to learn about the phenotypic and the immunologic and the genetic. And y'all, if it is genetic, it has to deal with DNA or RNA directly, like deliberately. We are testing for those nucleic acids, period. And the matching of them, the sequence of them is important. Immunologic tests are looking at antibody-antigen interactions, usually from the serum. So then we call that serology as well. So anytime you have antibody-antigen interactions to identify a disease, that is immunologic. If it is not immunologic, if it is not genetic sequence-based, it is probably phenotypic. Phenotypic is looking at what kind of media you can grow on. Did you make this byproduct? Did you change the media this color? Could you grow on this? And what was your stain, positive or negative for acid fast? And those kinds of things are phenotypic tests, all right? Some phenotypic tests we already learned about in our lab stuff. Those all in the second half of lab. Actually, everything we learned in lab was phenotypic tests, right? Except for ELISA. ELISA was the only test that we used that we actually had immunologic testing. So that dealt with antibody-antigen interaction. Everything else we did in lab was phenotypic. And again, there's other ones that are immunological that we didn't use, like Western blot or immunofluorescence. But again, those are looking at antibody-antigen interactions. Western blot works a lot like the DNA gel electrophoresis. We're just looking at proteins moving in the gel and then using antibody-antigen interaction to those bands that we get separated out from the proteins to look to see if that protein that we're interested in is there or not. And that's really all Western blot is. Immunofluorescence, we're labeling the antibodies with a fluorescent probe to look for color, like glowing color, literally, when you shine lasers at it. Oh my gosh, with specificity and selectivity, sensitivity is what that's supposed to say. Sensitivity and specificity. Okay, sensitivity, if you're really, really sensitive, you're going to pick up on anything and be bothered by any little thing. You're going to see all of them, all the positives. If it's there, man, I will know. I can be on the freaking other side of the house and hear my freaking dogs crying. And I'm so sensitive to that and I get annoyed by that. But I will hear every dog cry that there is. I see all the positives, right? If you're selective, then basically what you're going to be doing is you're only going to see the positives. So I might at some point get annoyed and hear every little creak in the house and think it's a dog crying. Then I might be very sensitive, but I'm not very selective then, right? But if I only hear dog crying, then that is specifically only for the dog crying, as well as very sensitive to it. And so those are the difference kind of between those two. You want to have both, obviously. You want to be both of those things. But that's the idea with those. I also want to say indirect versus direct testing with our immunological tests. Indirect, you're looking for the antibody. Has the patient ever had an antibody made against the thing? Has the patient ever seen the thing? Versus direct, does the patient have the thing in them right now? That's really the questions that you're asking with these, right? With whether your antigen is present or whether the antibody is present. I think that's pretty much it. Yeah. So that's all I have for units 1 through 3 for our like big picture review on all these to refresh you guys' memories on these things. Does anybody have any questions for me for units 1 through 3? I will be posting the recording for the other chapters. Again, I'm sorry that I was on the weird link. I literally didn't even know that it kicked me to a different link. So that was a surprise. I'm glad that I noticed it eventually and we were able to get some of you guys on here. But yeah, if you guys don't have any questions about units 1 through 3 review here, and I'll post chapters 23 and 24 walkthrough just as soon as I get off of this. And if you guys have any questions while you are studying, please email me with your questions if you can't think of any right now. Otherwise, I'll post this review. I will not be doing a separate review for Unit 4. That's a lot of memorization type stuff. I'm not sure that my reviewing it is really going to help anybody outside of that, especially with so much stuff that you guys have to know for the final. So we will be playing on Sunday with the Kahoots. I have a Unit 1 and 2 Kahoot that we will have questions for that one that we'll play through for like an hour. And then we'll play through the Unit 3 and 4 Kahoot questions set for another hour after that. So it'll be from 3 to 4, we'll do Units 1 and 2. And then from 4 to 5, we will do Units 3 and 4. So Unit 4 will be included in that on Sunday. Again, at 3 p.m. we'll start that. Any more questions from you guys?
I do.
Yes.
So I have to work on Sunday. I go in at 3. Will I be able to access the review Kahoots
Yes,
when I
I'll
get
post
home?
those separately. You'll have to
Okay.
like just go through them manually. You know what I mean? Like you'll get to
Yeah.
see the questions, but you can't play the games. But yeah, I'll make those available for sure.
Okay. Okay.
Anybody else have any questions? All right. Well, if that is it, I have really got to pee. I'm not even gonna lie. So, so I'm gonna go ahead and head out. Like I said, I'll post this. I'll post the one that nobody got to see from chapters 23 and 24 as soon as I can. Again, you didn't really miss a lot. If you can look through the slides, you really pretty much get it. I didn't talk really about anything outside of it. Then we'll have a review on Sunday at 3 p.m. And I hope I will see everybody there. I will be there on the correct link. And believe me, I'll make sure that it's working correctly. For that one, wouldn't make any sense for me to do it by myself. So, yay. We're almost there, guys. All right. So I'll see you guys on Sunday, I hope. And please get all your evals done, get your quizzes and everything done, get that out of the way. And we're almost there.